Good morning, everyone. I'm going to get us started today, and as people join the waiting room, we'll kind of let them in, and we're just going to get started right away so we can get through this highly anticipated presentation. So this is with CureDuchenne and REGENXBIO. As you might know, we're going to be talking about RGX-202, which is their investigational gene therapy for the treatment of Duchenne muscular dystrophy. We're so grateful to be able to be on the line with a bunch of team members from REGENX. It's been a pleasure to work with them and to release this information to the Duchenne community. I know there's a lot of excitement surrounding this clinical trial and this program. So this webinar is being recorded, and it will be available to view. Hi, Olivier. We have Olivier from REGENX on. Nice to see you. So this webinar is being recorded, and we'll be able to view it after on the CureDuchenne website. So in the event you want to share it along to a friend or family member, we'll send you that link to be able to do so. There is a Q&A tool that we're going to be using. It's at the bottom of your screen, and would like for you to kind of use this throughout the webinar. Even though we're going to answer questions in about the last 15 minutes, when something does come to your mind, don't be afraid to toss it in the Q&A box. We don't want you to forget it or forget the details of it. And then anything additional, if you come up with something 5 minutes after the webinar ends, we are always here to answer your questions. So that email is cares@cureduchenne.org, and we will forward that on to the appropriate team members at REGENXBIO to get you a specific answer to your question.
Thank you.
Yeah. And just a few more updates from the CureDuchenne side. I would be remiss if I didn't mention our upcoming conference that we're so excited about. It's in San Diego this year. April 20th is the welcome reception, but conference activity is the 21st through 23rd. You can go to cureduchenne.org/futures for more information on that. And of course, we're right in January, so this is the very beginning of our year, and this is our CureDuchenne events calendar. So we're going to a bunch of different cities this year for both workshops and family dinners. We call them sessions. We would absolutely love to see you. Those are up on cureduchenne.org/events. And we're really looking forward to a full year of being back in person and engaged with people. All right, next up, I'd love to pass the presentation over to Dr. Dastgir and Dr. Veerapandiyan, and Olivier as well. They could join me. Excellent.
Thank you so much.
Yeah, absolutely. I'm going to go ahead and go on mute and turn my camera off, but I'll be here for any logistical needs if you have them.
Okay, thanks. And I think we have Dr. Veerapandiyan as well. That's great. Okay, so thank you so much to all of you for joining today. We are so grateful for your time and attention. Today, we're going to be talking about RGX-202, which is REGENXBIO's investigational gene therapy for the treatment of Duchenne muscular dystrophy. I'm Jahannaz Dastgir. I am the Senior Medical Director and Clinical Development Lead of the RGX-202 program at REGENXBIO. And we have Dr. Veerapandiyan, who is a pediatric neurologist and co-director of the MDA Center at Arkansas Children's. He's a neuromuscular specialist as well. We can move on to the next slide. These are our company's forward-looking statements, which we have to present, and then we'll move on to the next slide. So this is me, and we can go on to the next slide. And this is Dr. Veerapandiyan. He is the director of the Comprehensive Neuromuscular Program, which is also a PPMD certified Duchenne Care Center and co-director of the MDA Association Care Center at Arkansas Children's Hospital. Just to give you some disclosures for Dr. Veerapandiyan, he has recently received compensation for ad hoc advisory boards consulting activity with Biogen, Novartis, Avexis, Sarepta, PTC, Scholar Rock, FibroGen, AMO Pharma, Pfizer, Muscular Dystrophy Association, PPMD, and France Foundation. Next slide. So let me tell you a little bit about our company, REGENXBIO. REGENX is seeking to improve lives through the curative potential of gene therapy. We are a leader in AAV gene therapy with exclusive rights to the NAV technology platform. We have a broad internal pipeline with expertise across multiple therapeutic areas, including neuromuscular disorders. We have end-to-end capabilities from starting with research through clinical development and then commercial-ready manufacturing. Our proprietary AAV manufacturing is known as the NAV Express platform, and we have internal manufacturing facilities in Rockville, Maryland. For our investigational gene therapy for Duchenne, we are seeking to advance a novel gene therapy to improve the lives of boys with Duchenne. RGX-202 is an optimized microdystrophin gene therapy with unique features. REGENXBIO is partnering with you, the Duchenne community, to help guide development of our program. And we currently have a Phase I/II study known as AFFINITY DUCHENNE that is now active and recruiting participants. On the next slide, I'll tell you a little bit about RGX-202. So RGX-202 is a microdystrophin designed to retain key elements of full-length dystrophin. So on the left here, you can see a picture of the full-length dystrophin a full-length dystrophin protein. Dystrophin is the protein that is coded for by your gene for DMD. And basically what happens is there's some anomalies in there that could lead to Duchenne muscular dystrophy. This is the protein as it exists and as it attaches to the sarcolemma or the muscle membrane. You can see that it is a long protein, what's listed here in kind of the blue array of circles, and it attaches to the sarcolemma and multiple other membranes in the sarcolemma. Unfortunately, the dystrophin gene is very large. So all gene therapy programs right now have to have an abbreviated form of the dystrophin protein known as microdystrophin. What differentiates our microdystrophin from other companies is that we have a unique structure that includes the C-terminal domain. You can see that here, that is point to as CT domain. This CT domain binds to other proteins that then also support an additional anchor to the sarcolemma. And we're hoping that with our clinical trials, we can prove that this construct or microdystrophin that we'll give you with this RGX-202 will be a stable and successful therapy for kids with Duchenne muscular dystrophy. On the next slide, I'll tell you a little bit about preclinical evidence in the DMD animal model. So I'm sure most of you know that the animal model that is typically used for Duchenne muscular dystrophy is known as the MDX mouse. So this is our data from the trials involving the MDX mouse. Here you can see the percent of microdystrophin expression in the individual muscles tested in the mouse. So the gastrocnemius is a leg muscle, the diaphragm is the breathing muscle, and of course we have the heart. With the different colors, you can see that there are different doses that are administered. The different doses are escalating, with black being no dose given, and purple being the highest dose given. And with higher doses of RGX-202, you can see higher expression of microdystrophin levels. We have an animation on this slide, if you could click again, sorry. Great. So what we also do in the mice is that you have to test the individual muscles and their kind of strength or force. And so again, you can see that there's a significant increase in muscle function and strength with the increasing doses of RGX-202. And finally, on the bottom section with the pictures of muscle fibers, these are pathology stains. You can see here that there is in the no-dose patient, in the no-dose mouse, there's all of this extra blue staining. This extra blue staining points to fibrosis within the muscle tissue. And fibrosis is basically scar tissue, or what we call fibrofatty replacement. So you can see with a mouse not receiving RGX-202, there's a lot of that blue tissue there. With the lowest dose that we've demonstrated here with RGX-202, that blueness has decreased. With dose 2, it's gotten even less. With dose 3 and dose 4, it's even less. Dose 4 almost looking normal. So we're hoping that these findings that we've seen in the MDX mouse are now going to translate into our human studies. Next slide. So how do we do this? So first you have to create RGX-202. So RGX-202 is an adeno-associated virus vector delivered gene therapy. So you've probably heard about all the different adeno-associated viruses that are used for gene therapy. Ours is AAV8. It's an AAV8 vector. We are using something called an SPC512 muscle-specific promoter, which will help it get targeted appropriately to the muscle tissue. And we have an optimized microdystrophin that again uniquely contains that C-terminal domain. After developing that, we moved to the preclinical or IND enabling activities. So the preclinical studies involved MDX mice and non-human primates to support transition to the clinic, to support moving into a clinical trial with our boys with Duchenne. We have manufacturing process development, provider and community input, and a clinical study design that's in place. Check. So now where are we? We're so excited to say that we are now active in recruiting patients for our Phase I/II clinical trial known as AFFINITY Duchenne. Thanks to all the community engagement, this has been a process to get here, but we are so excited to be here and to support the Duchenne community. On our next slide, I'm going to show you a little bit more of a schematic on what RGX-202 looks like. So it is a gene therapy that is set to address the lack of functional dystrophin. So we have the AAV8 vector, which delivers the transgene to skeletal and heart muscles in the body. We have the promoter, SPC512. This is a muscle-specific promoter that drives the expression of the protein in muscles. And we have the microdystrophin gene, which encodes a novel protein designed to retain key elements of full-length dystrophin. Again, uniquely including the C-terminal domain. This is a one-time IV infusion of drug. So now we're going to move on to information about the AFFINITY Duchenne Phase I/II trial. I'm going to pass the baton to Dr. Veerapandiyan so he could take it from here.
Thank you, Naz, and thank you for reading my disclosure slide. That was a long one. Thank you. And thanks everyone for joining. I'm Veerapandiyan. I'm a pediatric neuromuscular specialist at Arkansas Children's, and I'm also one of the investigators for this trial, and I do not work for REGENXBIO. Let's briefly kind of talk about the study. So it's an open label, multi-center trial, meaning, it's going to be multiple sites and it's open label. There's no placebo control or anything like that. It is Phase I/II, mainly looking at the safety of this gene therapy and also tolerability. And some of the key eligibility criteria, we are primarily looking at boys between age 4 and 11 years old who have to have genetically confirmed Duchenne muscular dystrophy, and the mutations has to involve exons 18 and above. So we are excluding boys that have mutations from exons 1 to 17. And these boys have to be ambulatory, and they should be able to perform 100-meter walk without any assistive devices. And obviously, like you all know, they have to have the antibody testing, and they should not be having any pre-existing antibodies to the AAV8 capsid. And of course, they should not be on any other investigational product or commercial gene therapy. In terms of the outcome measures, of course, like I said before, the primary outcome is safety. So we're looking at the safety assessments throughout the study. And we're also looking at efficacy, mainly the strength and function measured by some of the assessments that we normally do in these boys, like North Star ambulatory assessment and other assessments. And there's also going to be a muscle biopsy looking at the microdystrophin expression, expression in the muscle fibers, and also impact on the skeletal muscle and heart muscle through the MRI. Next slide, please. So this slide kind of talks about the overview, a brief overview of the study. Of course, when we bring these boys, they undergo the screening process to make sure that they meet all the inclusion and the exclusion criteria, the eligibility criteria for the study. Once they go past that stage, if they're eligible, then of course they get the intravenous infusion, and we will continue to follow them throughout, mainly for safety and tolerability throughout the study. And at 3 months, these boys get muscle biopsy to look at the microdystrophin expression. And as you could see here, 6 months, 9 months, and up to 12 months, again, continue to follow for them to mainly look at the safety and the tolerability of this product. And that's fine. So in this trial, again, we are using a comprehensive immunosuppression regimen. Like, you all know that different trials use different immunosuppression, like additional steroids and other agents to mitigate or to prevent the immune-related side effects that can happen with the gene therapy. So in this study, we are using a more comprehensive immunosuppression regimen, attacking different different parts of the immune system. And if you look at that, since this is a phase 1 and a safety study, in terms of the enrollment, it is going to be a stepwise enrollment. I'm gonna, if you could look at this picture down here. So the first subject that we're gonna enroll has to, of course, we want to start with a low dose and also low body weight, so they get lower amount of the viral vector. Again, we are looking at the safety, and most of these side effects are related to the immune related side effects to the immune reaction to the vector. So the first subject is going to be, of course, less than 20 kilograms, and they'll get this here's a dose, 1 to 10 times to the power of 14, and we will follow the safety for this patient, for the subject or the boy who's enrolled. And the next subject in this cohort would be less than 30 kilograms, and the next one would be less than 40 kilograms in this cohort. And these first 3 subjects would be dosed at least 4 weeks apart. And once that's completed, this will be looked at, that the safety data will be looked at by the safety data monitoring committee, and then we would escalate the dose to the next cohort, which is right here, 2 times 10 to the power of 14. And again, they would also undergo the same stepwise enrollment based on the weight, and just to make sure that we are starting with the lower vital lower. So less than 20, less than 30, and less than 40 kilograms. And then once that's done, then these cohorts will be expanded to include other subjects. Next slide, please. And now I'm going to pass it back to Naz to talk about this Affinity Beyond study.
Thank you so much for that. Thank you. So sure, we have a lot of questions coming through that we will address at the end of the presentation. What I want to talk to you about now is our Affinity Beyond study. So this is an anti-AAV8 antibody assessment study of boys with Duchenne muscular dystrophy. So this is a spinoff. This is something that we thought would be helpful for our population. And hopefully you'll find it something that you're interested in participating in. It is a non-interventional study seeking to understand the prevalence of AAV8 antibodies in boys with Duchenne and identify those who may be eligible for Affinity Duchenne. So that's the trial that we were just speaking of, and other future trials for those interested. So this is speaking to AAV8 antibodies, because if you have antibodies to the vector, unfortunately, the gene therapy will most likely not work for you. So we are excluding individuals who are positive for AAV8 antibodies. This is a study that will help us identify whether or not you are AAV8 positive and whether or not you could potentially be eligible for our phase 1/2 study or Affinity Duchenne. Participation consists of a phone or video interview and a home health visit. So we have a central PI, central primary investigator, who will do consenting with you over the phone. And once we find out that you're eligible for the study, we can send a nurse to your home. It's the convenience of your home to get the blood work done in order to check for the presence of the antibodies. We seek to enroll up to 200 participants in the United States. And our key eligibility includes boys between 0 to 11 years of age. Now, I saw one of the questions, I snuck a peek there and I saw, is it up to 12 or 12 is not included, but like 11 and 364 days is. So that's what we're talking about. So once you hit your 12th birthday, you are excluded. And that's the same for Affinity Beyond. The reason why we're opening up the antibody study to boys that are younger is because we know it's going to take time to get these results back. And we're hoping to expand the study in the future to hopefully do like a phase 3 study. And then these patients might be eligible. Of course, things might change with the protocol and the ages of inclusion. So we're trying to kind of aim for the future by enrolling kids who are a little bit younger. You must also have a diagnosis of Duchenne muscular dystrophy, and you must have not received any prior gene therapy product because that can alter the results as well. So on the next slide, we're going to give you a little summary of our presentation. So RGX-202 is an optimized investigational microdystrophin gene therapy with unique features. REGENXBIO's phase 1/2 Affinity Duchenne study is active and recruiting, with the first US site open with Dr. Veerapandiyan at Arkansas Children's in Little Rock. REGENXBIO is partnering with you, the Duchenne community, to help guide development. We always appreciate your feedback at any part of this process. Things can change based on what you offer to us in terms of information and concern. So we always welcome you communicating with us. We're a partner with you. And AFFINITY BEYOND is a non-interventional study primarily seeking to study the prevalence of AAV8 antibodies and identify individuals who may be eligible for the AFFINITY Duchenne or future studies we have at REGENXBIO. So thank you so much. So if you would like to learn more about our company, our website is www.regenxbio.com. We have an email that you can reach out to us. We have a lovely patient advocacy team who's also on this call, who's womaning that email, Duchenne@regenxbio.com. And we have clinicaltrials.gov listings for AFFINITY Beyond and AFFINITY Duchenne that are listed on this slide. So thank you for your attention. So I believe Carrie is going to be taking over the Q&A session, or maybe Liana.
It's Liana.
Liana, sorry. Sorry.
Hi, and I think I'll have my colleague Mike join us as well. Thank you so much for your presentation. It was very clear, and we have a number of questions that'll be coming in. Just to start with some of the logistics, someone asked whether the recording for the webinar will be, it is being recorded and it should be available in the next day or two. And you can find that through our website. And I'm just looking through the questions. So maybe we can start with the clinical trial and the inclusion and exclusion criteria. So one being, you addressed great, Naz, thank you very much, that up to 11 years old means they can be 11 plus some months, but doesn't necessarily means they have to be after 11, younger than 11. But there's a lot of questions about the exclusion criteria, about excluding individuals who have mutations in the first 17 exons of dystrophin. And maybe you can talk a little bit about why that is the case.
Yeah, this is a terrific question. So basically, the reason why we are being super cautious with this is because there were some adverse events noted in other clinical trials. This is our first in human trial, so we don't have any data of our own to go off of in terms of our drug product in humans, but we are being very cautious based on the foundation that was put before us and the concerns and the consortium that's been in place, headed by Dr. Bonneman and Dr. Montoni, to kind of make sure that everybody's proceeding appropriately with the appropriate precautions in place. And basically, in some of the other studies, there had been some very severe adverse events, and they tied those adverse events to individuals who had mutations in the 0 to 17 range. Because this is our first in human study, we are going to test the safety of our drug in those who do not seem to be predisposed to serious adverse events. And then we do hope to then open it up. Once we establish safety, we do hope to open it up to other exon mutations.
Thank you very much for that. And, one thing that I can add is that this has been a conversation that's been going on for over a year. With all of the gene therapy clinical trials and all of the companies involved in that. And so this is of a big concern, and the companies are working together to try to understand it and try to figure out the strategies to get over the limitations of it. And so, just from a more general perspective in the Duchenne community, we recognize that it's always disappointing to hear when there's some individuals who can't participate, but this is being actively worked on, not just by REGENX, but lots of scientists in the field. And so I just wanted to make that point for the families as well.
Yeah, could I just expand a little bit? And first of all, it's great to see REGENX active in the clinic. We welcome you, and it's always fantastic to see a slightly different variation on the gene here. And I think it's truly going to be interesting to see how this plays out over time and what advantages it has. Could you just talk a little bit about your future plans for non-ambulant patients? We tend to sort of neglect at times some of the older patients. And I think particularly some of your thoughts around going into much younger patients, younger than 4, where we think the clinical benefits may be quite profound.
Sure. So to address the non-ambulatory patients, so basically non-ambulatory is typically an older patients, and older patients typically have higher weights. So we would love to expand in the future to potentially include these patients. But for now, again, from a safety standpoint and the goal for a lower viral load delivered, because we're doing per kilogram dosing, we're aiming with the younger patients. So that's why we have not addressed the non-ambulatory patients just yet. That doesn't mean we're not thinking about it. It's always on my mind, always on our mind as a company. The other limitation of non-ambulatory patients is also establishing good outcome measures that can be accurately tracked over time to show responsiveness to the drug. So typically we do that with the younger patients who can perform some of the outcome measures that require ambulation. And then if we can establish that that works in conjunction with some other outcome measures that we can use in non-ambulatory patients, then we can then hopefully successfully prove that it's helpful for non-ambulatory patients as well.
Well, thank you for that.
With regards to the younger patients, it's almost a similar issue in the sense that there aren't really great outcome measures for babies. So that is a little limiting, although we are really actively working on trying to establish potential ideas for addressing the younger population as well. But the outcome measures are what makes it difficult. So it's also an ambulation issue. And also because Duchenne's natural history is such that you might not see, I mean, you do see subtleties if you're very used to seeing it, but do the outcome measures actually capture the subtle changes in gait that you see in a toddler? It can be very difficult to decipher that. So that's what we're working on. Again, constantly thinking about it. We're always thinking about the future of this Duchenne program that we have and how it's going to evolve. So it's always on our mind.
Thank you. Is there a weight cutoff for inclusion into the ambulatory part of the trial?
So there's not an ultimate weight cutoff. What we are doing is we are dosing in a stepwise fashion with an escalating weight.
Okay, thank you.
Once we establish safety at less than 40 kilos, we hope to go beyond.
Okay. The community has been very familiar with AAV9 and RH74 that's been used by Sarepta, Pfizer, Solid. AAV8's new to many people. Is there a difference or an advantage to AAV8?
So AAV8, I think, basically is very good at targeting muscle and heart, as we mentioned before. I would probably put it as being a bit more similar in function to AAV RH74 than AAV9. We're hoping that it's going to be less immunogenic and more effective. So
Okay.
that's how that was chosen.
Thank you for that.
Mm-hmm.
Leanna?
Sure. Switching topics a little bit, there's a lot of questions around the dosing and sort of what you saw preclinically and, what levels of dystrophin you saw there, and in terms of how that compared to other programs that some of our attendees may have been used to seeing from other companies, and then how that informed the dosing regimen for your trial.
So, in terms of how it compares to other programs, I can't really speak to it without having a head-to-head comparison available to see. In terms of translation of the dosing, I know we have Olivier on the call. We established a certain protocol with the mice, but then taking that dosing and translating it into human, I know was a decision that was made by the bioanalytical group, and also basically doing mouse and non-human primate studies to establish safety in them. But I don't know if Olivier had any other comments on that issue.
Yeah, my comment on that is that there is something very specific to gene therapy, which is that we have to obviously study safety first, but since this is a permanent treatment, it's difficult to propose to the patient to have a very low dose that we know is going to be inefficient, right? This is just not happening. So we're doing everything we can to find a lower dose, which is already efficient and has shown to be efficient in our preclinical studies. It may not be the optimal dose that we'll eventually want to have with many patients, but this is a dose which is very reasonably efficient, and we're just asking whether this first dose is safe, right? So I mean, this is with this in mind that we've designed the trial, and this is based on very, very careful assessment preclinically of what a dose increase makes in the animals. When do you start to see an effect? When do you plateau? And it doesn't make sense to use more vector. So our goal here is really safety to start with, safety, but those are doses that are likely to also bring benefit to the patients. And so this is the complicated mix that we're trying to have here. But given our preclinical data, we're very hopeful that this should be what we will observe in the clinic with the patients.
Thank you. Could you just comment a little bit further, a bit more details about the biopsies? I presume there's a pre-dose, and I've seen one at 3 months. Was there further biopsies, and what type of biopsy are you taking here?
A great question. So the first 3 patients of each cohort, this was established with a discussion with the FDA, will receive muscle biopsies. These, you will have one prior to entering, obviously, into the study to have a baseline. And when we check for microdystrophin expression, this will be done at 3 months. So it's going to be an open muscle biopsy, which we've seen, and we've primarily decided upon that because that is the cleanest sampling of muscle tissue that we can get. So that will be performed in that way at that time, those time points.
Okay. Another point, just to clarify for me, and I seen a question from Tushar on there about it, but the first patient was going to be less than 20 kilograms, the third less than 30, the third patient was going to be less than 40 kilograms. Was that right?
Yes.
Okay, got it. Thank you. One other one that's come up, I think consistency a couple of times here, is the number of sites, what you're planning for. I know we want to push you further than where you are today, and you may not know the answer to it, but we know that Arkansas is active. What's the company's plans going forward into this year?
Yeah, for this year, we plan to open up additional sites in the United States in locations that are convenient. So we have Arkansas in the south. We have other sites on the East Coast, Middle America, and West Coast that are not yet active, but we're getting them up and active. And so once they are, we'll put them on clinicaltrials.gov so you'll know where you can go to.
Excellent. Thank you.
And along those lines, is this going to be US based only, or is there plans to go outside of the US as well?
Yep, we have plans to go outside the US, but obviously it's a process. So we have plans to go into both Canada and Europe.
Thank you.
Mm-hmm.
So there's some just general questions here about gene therapy. And I think, Olivier, you sort of mentioned one of them that, at the moment, there's only an opportunity to be dosed once with an AAV. Because individuals develop antibodies, but there's just sort of general questions in terms of what are sort of the risk factors or some of the considerations when considering gene therapy. Maybe you can just speak a little bit more simply to that.
So you're asking about the risk factors here. We've seen events in the clinic in previous trials occurring with very high doses of AAVs, and those now have been analyzed. There starts to be some understanding of what's going on, although it's not fully understood. But actually, it really guides the way we're very carefully increasing the dose and starting with where we start. So this is we also. I mean, the field is working at improving the AAVs themselves. I mean, we now have access to capsids of AAV, types of AAVs that may be a bit more efficient at targeting the muscle. They're being developed. They're not available yet for clinical trials. It will still take a couple of years. So that's one area where progress is being made. And so, again, this is being developed. So I can't really promise anything, but we're very hopeful that we can get better products that will be administered at lower doses and therefore have less toxicities. And so again, the one-off gene therapy is what we all hope for, but this may be asking a lot. There are many issues associated with that. You treat a patient which is still young and will then grow, and you may dilute the effect of your gene therapy as the muscle grows in size. And this is something that we still need to figure out the real effect of this in patients. So are we going to be? I mean, are we going to have an effect that lasts for 5 years, 10 years, 15 years? This is something that is still out there, right? And we want to be very, very cautious. We don't want to overpromise. I think discussions with patient communities really indicate that even a therapy that would bring a benefit for 5 to 10 years is something that's already perceived as enormous, right? So worth doing. Can we re-administer? Can we come back with another gene therapy? It's not out. I mean, it's again, the field is working at ways that we can develop to be able to do that, and this is about teaching your immune system how not to overreact to a second injection of gene therapy. So there are, thoughts and experiments and actually data showing that you may be able to do these kind of things. Again, this is not ready for prime time. This will take a few years before we can bring this eventually to the clinic. But we remain, hopeful that something will be done in this direction.
Thank you, Oliver. Yeah, I was going to ask you just to extend that just a little bit, because it reflects on a couple of the questions that families were asking. Has the company got any information on the cross-reactivity of neutralizing antibodies to AAV8 versus RH74 or AAV9? And I think what parents think about is, for those children that perhaps failed to get entry into an AAV9 clinical trial because antibodies may have been high, is there an opportunity here that they would not cross-react with AAV8?
There is certainly an opportunity, but there is also an overlap, a gray zone where people will react to both, right? So it's really that the reaction against the capsid doesn't target just one particular site on the capsid, but multiple sites. So it gets complicated, and some individuals will have high immunity against AAV9 and no immunity against AAV8, or individuals will have high against AAV9 and some against AAV8 that will still exclude them, right? So that's and our current trial on immunity around AAV8 will bring us data to understand this more, actually. But today it's difficult to make a prediction for each individual on how they're going to cross-react and react at towards different AAVs.
Thank you.
Yeah, and just to thank you, Mike, for that. And to expand on that, there's other questions about, could being in this trial make individuals ineligible for some future treatments? And I think, as you've said, Olivier, at the moment, we don't have the opportunity to redose, right, with another AAV8 or potentially AAV9. Yeah. But in terms of other treatments that are not gene therapy, maybe you can speak to that.
Sorry, can you repeat your question?
Yeah, so there's questions about whether or not getting this treatment would make individuals ineligible for some future treatments. And, I think you can sort of pool them, right? There's the gene therapy, AAV delivered, and then there's things like steroid, like the other types of therapies.
Yes. So not necessarily, although this is going to be a case by case, but people who've been treated by gene therapy don't necessarily lose the ability to receive the other treatment systematically. I mean, that's not the case. But I want to be careful here. I mean, each case is going to be special. Maybe the gene therapy can bring you an immunity against the dystrophy that you didn't have before, and then it will be a problem for other treatments that would reactivate dystrophin, like the exon skipping, But again, maybe not. So it will be individual, and we're going to have to be careful about it, of course. But there is no. It's not that if you opt for gene therapy, this is it, and you won't ever have anything else. Not at all.
And maybe this is, I can open up this question to everyone. You know, do we expect individuals who have received gene therapy to continue to take steroids or other sorts of medications?
Yeah, this is an inclusion criteria for the trial is that you have to remain on your home dose of steroids in order to enter the trial, and you'll be on that course of steroids throughout the duration of the trial.
I think, Leona, just kind of piggyback on both Olivier and Naz. If the question is after the trial is done, are they going to be still on steroids? Are they going to drop steroids? We don't know yet. We don't have any information on that yet, right? So I think that's also another discussion going on in the community, how that's going to work. It's going to depend on a lot of factors, how soon they are treated, and all of these trials that we have data, all of these boys are on steroids already. They haven't stopped steroids. So I think we don't know answer for that. And also kind of going back to this question that Olivier kind of touched upon in terms of, future treatments. And I think if we're talking about non-gene therapy clinical trials, that's also going to depend on how that trial inclusion exclusion is going to come up with. They may exclude anyone who received gene therapy. We don't know. So that's going to depend on those sponsors and the company, that trial's inclusion exclusion. But if you're thinking about hopefully we have some non-gene therapy treatment approved at the time other than steroids, then I would assume that they have to complete this study, and then it's going to be commercially depending on how they discuss that with their own physician and come up with that treatment plan.
Mm-hmm.
Terrific, thank you for that. I see a question here about when people are already so excited about this, when can we expect to see the first set of results maybe come out of the first few cohorts?
I mean, I think, obviously we'll be presenting data as we get it through the trial, as we can put things together at different meetings. So I'm sure patients that attend different meetings, once we have some reliable data to present to you, we will present it. We'll keep you updated on what's going on. But this study has a duration of 12 months. And obviously there's some analysis time that's required afterwards. This particular part of the study, we do have a long-term follow-up study that also follows the patients for 5 years. But I think after we can analyze the first 12 months of data, we can easily publish this information.
Thanks. And
obviously we can be a little bit more precise once we actually dose our first patient, which is something that should happen anytime now. But then we start the clock, and we know that 12 months later we have data, and a few months later we can discuss this data publicly. So that's the time frame.
And I'll just go on record saying, I'm sure, our CARES team would love to have you, when you have that data, come here and discuss it in detail with all of us.
Yes, it will be our pleasure to do so.
Yes, definitely.
So I'm scrolling through the rest of the questions. I feel like we've addressed just about everything. Is there anything else you see, Mike?
No, I think that we caught them all. I was running through them in real time as well.
Yeah, I think there was a lot of really great questions here, and I think that just speaks to, first of all, how important therapies like this are to our community, and just how detailed their understanding is. And so, again, it's thrilled to have you guys starting your trial, and we look forward to more information in the future.
Absolutely.
Great.
Thanks so much to the entire REGENXBIO team. Thanks to Dr. Veerapandiyan, and thanks to Liana and Mike for facilitating. This is a really engaging Q&A. I also wanted to let everyone know that in the event that you felt as though you have follow-up questions, or maybe you need a little bit of clarity on your question, please do reach out to me at cares@CureDuchenne.org. With the presence of new clinical trials opening, we also do understand that there is kind of a burden on some families as they try to navigate this and make decisions and come up to speed on all these new clinical trials and what that means for their family. We run a one-to-one program. So myself, Mike, and Liana really enjoy getting on a Zoom call one-to-one with families and just helping you work through that information and empowering you to make decisions for your family, making sure that you have all the information or linking you to someone, at REGENXBIO who could helpfully answer your questions. But that is going to be it for today. Again, reach out if you need to, but thanks everyone. And that's the conclusion of today's webinar.